Anyone who says that men can't lactate has never seen a man doing intense exercise. Or maybe it just depends on what your definition of lactate is.
Ketogenic diet has neurological benefits. Why do we have to eat such an enormous amount of food?
Complex science, clear explanations. Class is starting now.
Hi, I'm Dr. Chris Masterjohn of chrismasterjohnphd.com, and you're watching Masterclass with Masterjohn. We are now in our 15th in a series of lessons on the system of energy metabolism, and we're continuing to make our way through our comparison of fat versus carbs to discuss the more complicated topic of protein later. And we've seen so far that because glycolysis breaks glucose down into pyruvate, which has a carboxyl group, owing to the extra amount of atomic oxygen within the glucose molecule, the decarboxylation of pyruvate generates one carbon dioxide that is not generated by fat metabolism, and therefore carbohydrates generate more carbon dioxide than fat. But pyruvate is also significant because it has several metabolic fates besides generating acetyl-CoA. And these are metabolic fates that fat doesn't have. It can be turned into lactate, which is the topic of today's lesson, or it can be turned into oxaloacetate, which is the topic 2 lessons from now. Later, we'll talk about how that ability to be turned into oxaloacetate under the conditions where it matters makes carbohydrates able to spare the need for protein in a way that fat can't. But now what we're focusing on is the ability of carbohydrates to generate lactate, something else that fat can't do. And that gives carbohydrate metabolism a special role under a variety of conditions. One of those conditions is in sports performance, where exercise intensity becomes so intense that we need to generate that lactate. However, there are other contexts where lactate generation is important besides sports performance. So we're going to break this into 2 lessons, and today we're going to lay down the basic foundation of the biochemical principles of why lactate is important and talk about the broad number of contexts in which that could play out. In the next lesson, we're going to take our deep dive into how carbohydrates affect sports performance at a very practical level. Shown on the screen is what we've already seen before, which is that glucose is broken down during a series of 10 reactions that occur in the cytosol, named glycolysis collectively, into 2 molecules of pyruvate. As an alternative to decarboxylation, which would produce acetyl-CoA, pyruvate can also be reduced to lactate. And when we say reduced, we mean it chemically. Electrons can be added to it. Remember, pyruvate is an alpha keto acid because it has a keto group on the carbon that's alpha to its carboxyl group. That keto group is electron deficient, hence the double bond. Therefore, it can be reduced to a hydroxyl group. And if you do that, you wind up with lactate. And lactate is an alpha hydroxy acid because the carbon that is alpha to the carboxylic acid part has a hydroxyl group. You may have heard of alpha hydroxy acids before because they're often added to cosmetics, and lactate could be one of the alpha hydroxy acids in some cosmetic preparations. Now, if you look at lactate, it has a special property where the carbon is chiral, which means that there's one carbon attached to it that's one thing, a hydrogen that's a different thing, a methyl group that's a different thing, and a hydroxyl group that's a different thing. And because everything is different going around it, you create a situation in which the orientation actually can make the molecule fundamentally different. And so when the OH is sticking out to the left side, that's not what L means, but it can be helpful to remember it that way. If you have the carboxyl group on top, when the OH is sticking out to the left side, that's L-lactate, distinguished by D-lactate. D-lactate is commonly noted to be produced by bacteria, often erroneously stated to be mostly a product of bacterial metabolism in the human body. That's not true. We'll talk later about where we get D-lactate from as a part of normal human metabolism. But for now, we're talking about L-lactate, and it's important to make that distinction because we're going to talk about D-lactate later. And the enzyme L-lactate dehydrogenase catalyzes the conversion of pyruvate to lactate, which is totally reversible. And in fact, it's called L-lactate dehydrogenase because it can oxidize and dehydrogenate L-lactate back to pyruvate. But if we're deriving pyruvate from glycolysis, then at that point, the direction we're going to be going in is NADH is going to reduce the keto group to the hydroxy group, producing L-lactate. In order to understand why the reduction of pyruvate to lactate is important physiologically, we need to start looking at some of the details of glycolysis. We're not going to look at them all right now, but we're going to look at the details that are important to the role of L-lactate in sports performance and in the other contexts in which it becomes important in the human body. The first step in glycolysis is the phosphorylation of glucose. This is both energy intensive and requires phosphate. So ATP contains energy and phosphate. The terminal phosphate bond is broken, and its energy and the phosphate itself are both included in the glucose molecule. What results is glucose 6-phosphate, and this is catalyzed by hexokinase. A kinase is an enzyme that phosphorylates, and hexo refers to the fact that glucose is a hexose, which is a 6-carbon sugar. The notable thing that happens for our purposes right now is that in the production of glucose 6-phosphate, the phosphate adds a negative charge to the glucose. So free glucose is not an ion. Glucose 6-phosphate is an ion, and that makes them fundamentally different. The panel on the left side of the screen shows the role of charges and polarity in regulating the ability of something to cross a cell membrane. If we take this lipid bilayer as the cell membrane, and here we're taking experimental data that just uses pure synthetic lipid bilayers to mimic the properties of the membrane without transporters in it. So if we just have a pure lipid bilayer with no transporters, what we'll find is that nonpolar molecules such as oxygen and carbon dioxide easily traverse the membrane. That's because if you look at this membrane, you'll see these little dots that stud each side, and those are the phosphate head groups of phospholipids. Phosphate is water-soluble and it faces the water-soluble portion of the cell, either the water-soluble outside or the water-soluble inside. The fatty acids are the tails in between. They're oriented towards the inside of the membrane, and they make up the vast bulk of the membrane. So to traverse the membrane is primarily to traverse something that's lipid. When we talk about polarity, we mean an electronegativity difference. If one atom holds on more tightly to electrons than the other, there will be a separation of charge. And because water is polar, water has oxygen that holds on to electrons much more tightly than hydrogen, then things that are polar will be very attracted to water because their partial negative charges will interact with the partial positive charges of the hydrogens of water, and their partial positive charges will interact with the partial negatives of the oxygen of water. So having polarity and having charge makes you very strongly attracted to water, and that water is going to hold you back from traversing through the lipid bilayer. In the case of oxygen and carbon dioxide, oxygen is totally nonpolar because what's the electronegativity difference between oxygen and oxygen? 0. Carbon dioxide is made of 2 polar bonds, but as we've seen outside of this slide many times during these lessons, the C is in the middle, the oxygens are on the 2 sides. So you have oxygen pulling electrons that way, and you have the other oxygen pulling electrons the equal and opposite way, with equal force in the opposite direction. And you wind up with carbon in the middle of a tug of war in which it's perfectly balanced because the negative charges are being driven in totally opposite directions. So the force of one oxygen cancels out the force of the other oxygen, and the carbon dioxide is totally nonpolar. So oxygen and carbon dioxide just make their way through the membrane like giving candy to a baby. Small polar molecules get through the membrane a little bit, but the majority of them don't cross. Water is a great example of a small polar molecule. Because of the polar distribution of charge, it, of course, is attracted to itself. Water is soluble in water. But because it's small, it's able to get through the membrane. And that goes back to the general principle that if you have an equal distribution of energy in the body, and you want to throw around things and try to make them do things that require energy, such as the energetically unfavorable mixing of water with lipid, then if what you have is small and it's low mass, it's easy to push around. And so because water is small, it's easy to do things that seem energetically unfavorable, like push it through the membrane. So when I say easy, I mean easy compared to other larger substances. It's still the case that water, for the most part, will not cross the membrane, but some water will. As you go up in size, you can take something like glucose. Glucose is polar like water, but it's much, much larger. So you get traces of free glucose that can cross the membrane, and the vast majority cannot. But when you get to charged ions, for example, hydrogen ions, chloride ions, sodium, potassium, phosphate, all of these things that have full positive charges or full negative charges cannot cross the membrane at all. It's the same principle of the partial charge of a polar molecule, only it's massively accelerated and amplified by the fact that you don't have partial charges, you have a full charge. And they are just too strongly attracted to water to cross a lipid membrane. Now, if we zero in on glucose, what we can see is that traces cross the membrane. That's a problem for 2 reasons. First of all, if you want to get glucose inside a cell, you need more than traces to come in. But second of all, if traces can come in, then traces can go out. And if you get glucose in the cell, you want to keep it there. So one of our solutions to that problem is to have a glucose transporter in the cell membrane. Glucose transporters allow the free flow of glucose into the cell and equally allow the free flow of glucose out of the cell. So let's say that you're exercising. You increase glucose transporters to get glucose into the muscle. Let's say that you eat a lot of carbs. The carbs go into your blood, your pancreas makes insulin and wants to clear glucose from the blood. Either way, that's achieved by increasing the number of glucose transporters in the membrane. But glucose transporters don't do anything to lock the glucose in the cell. They just allow the rate at which it crosses the membrane to massively increase. And if it massively increases so the glucose can come in the cell, it's going to massively increase going out of the cell as well. And if you're trying to concentrate glucose inside the cell, then you got a big problem because glucose is going to be traveling against its concentration gradient because glucose will accumulate here. It's going to get harder and harder and harder to have net transport of glucose into the cell. And so what happens is that when you phosphorylate glucose, you make it glucose 6-phosphate, and that negative charge on the phosphate does two things. First of all, making it a charged ion means that there's no transport of free glucose across the membrane. So these dotted arrows indicate that there's always transport of free glucose in trace amounts. That is totally shut off. But the second thing is glucose and glucose 6-phosphate have totally different chemical properties. So the glucose transporter recognizes free glucose and doesn't recognize glucose 6-phosphate as glucose. Because glucose 6-phosphate doesn't interact with the glucose transporter, then you have a situation in which if glucose is going to go in or out along its concentration gradient, then turning most of the glucose into glucose 6-phosphate maintains the free glucose concentration inside the cell very, very low. If you can maintain free glucose in the cell very, very low, then you maintain the energetic favorability of free glucose at high concentration outside the cell being transported into the cell where free glucose is scarce. So, two important roles of phosphorylation are one, to totally wipe out the trace transport of glucose across the membrane unassisted, and number two, to totally shut down its interaction with glucose transporters, maintaining low concentrations of free glucose in the cell and maintaining the energetic favorability of net transport of glucose from outside the cell to inside the cell. Now, it's important to note that phosphorylation doesn't commit glucose to glycolysis. It could have other fates. But when we're looking at glycolysis on its own, we would say that phosphorylation is the first step. But look at the overall objectives of glycolysis in this slide. We see that glucose has to be phosphorylated. Glucose is a hexose. When it becomes phosphorylated, it becomes a hexose phosphate. Pyruvate is oxidized and it's split in half. So first we're going to have to split the hexose phosphates in half to generate two triose phosphates. The phosphate continues to lock them inside the cell. The phosphate also plays other roles because it's acting as a reservoir for energy that's needed for some of those reactions. And as we'll talk about later, phosphates are actually protecting these molecules from forming potentially dangerous byproducts. And so when you get to two triose phosphates, you can't have only had the first phosphorylation. You need a second phosphorylation before you split them apart so that when you do split them apart, both trioses are phosphorylated. So in the first several steps of glycolysis, before we get to triose phosphates, we're going to have to invest 2 ATP. This is called the energy investment phase of glycolysis. After phosphorylation and cleavage to 2 triose phosphates, we have oxidation and dephosphorylation. And the oxidation and dephosphorylation are both going to release energy. We're going to wind up with 2-pyruvate, which has been oxidized, split apart, and dephosphorylated. In those later steps of glycolysis, we are not only dephosphorylating the 2 phosphates that we took in the first phase, but we're also releasing energy that's used to phosphorylate ADP beyond that. So the second phase of glycolysis is called the energy generation phase because we are producing 2 NADH and 4 ATP. But if we invest the energy from 2 ATP in the energy investment phase, and we release the energy in 4 ATP in the energy generation phase, then in net, as we produce 2 pyruvate from glucose, we are converting 2 ADP to 2 ATP. We're also converting 2 NAD plus to 2 NADH. Now, as you look at this general structure, this allows us to begin to see the role of lactate. Remember that when we convert pyruvate to lactate, we're actually using NADH to generate NAD plus as a product. If we look at glycolysis here, we see that in net, the reactants are glucose, ADP, and NAD plus. If we have a net production of 2 ATP and a net production of 2 NADH, that means that ADP and NAD plus are equally important as reactants in glycolysis. Now, let's imagine what would happen in the electron transport chain if oxygen became limiting. Ordinarily, we have NADH, FADH2, and oxygen as reactants along with ADP. Now, ADP is not generated in the electron transport chain. It's generated by other processes apart from cellular respiration that utilize ATP. Let's take as an illustrative example muscle doing work during exercise. If you contract your muscle more, you're going to use more ATP. That's got nothing to do directly with the electron transport chain. It has to do with the contractile proteins of muscle that use ATP for energy. Where it connects to the electron transport chain is now you have more ADP. That ADP needs to get phosphorylated in the electron transport chain. So if you keep doing muscular work, you don't need oxygen to do the work directly. You just need the stimulus from your nervous system, the release of signals in the muscle cell, and the contraction of the muscle protein to use the ATP and generate the ADP. But to convert the ADP back to ATP, in the electron transport chain, you do need oxygen. And doing that with oxygen is what generates NAD+. So right here, we start to see the interplay of ADP and NAD+, the 2 reactants we need for glycolysis. Now imagine a moderate amount of exercise, and you get a moderate amount of ADP. Well, what does that do? What it does is it comes over the electron transport chain, increases its activity, increases oxygen utilization, increases NAD+ generation. So usually, ATP utilization and the production of ADP and the production of NAD+ are all tied together, operating proportionally. And the reactions that fuel those in the citric acid cycle are also governed proportionally because of the production of NAD+ and FAD, and because of the allosteric regulation of the citric acid enzymes, the citric acid cycle enzymes that we've talked about before. And also glycolysis, which should increase when you have more NAD+ and more ADP, is also proportionally regulated. However, if ADP is produced by muscular work and NAD+ is produced in the electron transport chain utilizing oxygen, we can see now that they can be divorced from one another if the amount of work that is going on is so great that it exceeds the capacity of the electron transport chain. For example, if it exceeds the supply of oxygen. If muscular work outpaces the supply of oxygen, then more and more ADP is being made, but it can't be made into ATP in the electron transport chain, and it isn't generating proportional NAD+. You still have more ADP that consumes more oxygen and generates more NAD+. It's just that those latter aspects, NAD+ generation, are not keeping up with the degree of muscular work, the degree of ATP utilization, and the degree of ADP generation. So to summarize that, When you have oxygen supply that matches energy expenditure, then activity is going to increase ADP and is going to increase NAD plus proportionally. But when energy expenditure exceeds the oxygen supply, you get a much larger increase in ADP. And since you've maxed out your ability to generate NAD+, it still increases, but it doesn't increase anywhere near as much as ADP. During that situation, we have 2 ADP and 2 NAD+ that are equally important as reactants in net glycolysis. But ADP is in massive supply that's overwhelmingly disproportionate to the supply of NAD+. So 2 important things have happened. First of all, the reason ADP is accumulating is because it's not being phosphorylated in the electron transport chain enough to keep up with the demand. That means you need another way to generate ATP. Lo and behold, glycolysis could be your source of ATP, and yet you're missing NAD+ as a reactant. Do you have more when you're doing more muscular work? Yes. But do you have enough to keep up with the ADP? No. Now, in real time, what's going to happen is you have more ADP and you have more NAD+, and so they're going to speed up glycolysis. But ADP is going to accumulate much faster. It's a reactant, so it's going to speed up glycolysis. But as soon as you speed up glycolysis, NAD+ gets even more depleted because it's being converted to NADH. That can only happen for a very short time before NAD+ becomes extremely limiting for glycolysis. Right when you need glycolysis to provide the higher level of ATP that you need at the margins where the electron transport chain can't keep up. And now we have the context that we need to understand the physiological role of converting pyruvate to lactate. Our problem is that we don't have enough NAD+ to keep glycolysis running, right at the time when glycolysis becomes important to ATP production. So pyruvate consumes NADH, which is abundant, to convert it into NAD+ while making lactate. In that sense, lactate is just a means to an end of making NAD+ that you need for glycolysis. And it's really the NAD+ that's more important than the lactate. When we use glycolysis to make lactate, we call it anaerobic glycolysis. Really, the glycolysis part of it isn't different. We're still making glucose to pyruvate. It's what we do with the pyruvate that's different. And so in anaerobic glycolysis, anaerobic really refers to the context rather than having different reactions within glycolysis. Now, we've solved one problem, which is by making lactate, we can keep glycolysis running anaerobically. But we have another problem, and that's that if you look at the full oxidation of glucose to carbon dioxide, the net yield is 32 ATP. Some older sources will say 36 to 38 ATP. That information is outdated, and it appears to be 32 ATP on average. Now, if you produce lactate from glucose, you're only generating 2 ATP. You're generating 15 times less ATP than fully oxidizing glucose to carbon dioxide. That's a serious problem because you're in the crunch for ATP, and you're going to get a measly 2 ATP molecules out of this process. What kind of solution is that? Enter the Cori cycle. Although we only generate 2 ATP in muscle when we convert glucose to lactate, the lactate can leave into the blood, and it can go into the liver, and through gluconeogenesis, it can be converted back to glucose. Now, if glycolysis is energetically favorable and generates ATP, we can imagine, even though we haven't looked at the details yet, that gluconeogenesis is energetically unfavorable and consumes ATP. But in fact, gluconeogenesis is taking reactions that have extremely large negative delta Gs from glycolysis and trying to make its way around them. And it does that not by just reversing the reactions, because it can't, but by providing alternative means to get back to glucose. And by coming up with a convoluted way to go backwards into something energetically unfavorable, it actually has to invest more energy to convert lactate to glucose than is yielded by making glucose to lactate. So we are not breaking even here by any means. However, in the liver, you're not doing that much compared to what you're doing in the muscle. So if you're doing heavy, intense exercise work, it's your skeletal muscle that at that moment is the one that's operating above and beyond the oxygen supply. The liver, as the metabolic hub of the body, is prepared to help out in these circumstances, has the energy reserves available to help out, and is not being taxed with the work demand that skeletal muscle is in that moment. So the liver can afford to do something very expensive to support the energy needs of the muscle, as long as it's going to get paid back later. Now, if we look at what's happening here, I'm putting 6 ATP. We'll see later that some of this is mediated by guanosine triphosphate, which is GTP, but they're in a common pool with ATP. So it's the equivalent of 6 ATP being used here. The ATP never travels into the blood back and forth. So although you're not transporting any ATP to muscle tissue, what you are actually doing is taking the energy from the liver's ATP supply, investing it in the glucose molecule, and the glucose molecule is carrying that energy over to the muscle and releasing it into the muscle's ATP supply. Once it does that once, it can do that again, and it can do that again, and it can do that again, and again, and again. Every time it does that, you get more ATP out of that glucose molecule, and you have more net transfer of energy from the liver to the muscle, and you have greater and greater energetic debt accumulated in the liver. Now, if complete oxidation of glucose will yield 32 ATP, and anaerobic glycolysis only yields 2 ATP, you have a solution to that problem, because you can run this cycle 15 times and all of a sudden, boom, you've gotten your 30, give or take, ATP. 16 times you've gotten your 32 ATP out of that one glucose molecule. And that is the physiological significance of anaerobic glycolysis when tied to the Cori cycle. Now, anaerobic glycolysis is not just important to exercise performance. It's important in any condition where energy utilization exceeds the supply of oxygen. Some other examples are red blood cells. Red blood cells need to make room for hemoglobin, so they discard their mitochondria. If they don't have mitochondria, then it doesn't matter if they have oxygen, because they don't have the electron transport chain that uses it. So red blood cells always need to rely on anaerobic glycolysis to support their energy needs, which means they're always running on glucose. Some tissues are just poorly oxygenated. Because of their situation in the body, the oxygen doesn't reach them as well in adequate amounts to support their energy needs. And these include the lens and cornea of the eye, the kidney medulla, and the testes. These tissues are always running, to some degree, on glucose. Just as exercise increases energy demands, stress could also do that. For example, if you have the fight or flight response, you may release all this glucose and fatty acids to shunt it all towards your skeletal muscle in your brain so that you have the ability to powerfully contract your muscles and the mental acuity to either fight or flight. In that case, you have high energy metabolism, and if it temporarily exceeds the delivery of oxygen, just like in exercise, you can begin using a greater degree of anaerobic glycolysis. Suffocation is another example. Hopefully this doesn't happen to anyone, but it does. And if you don't get enough oxygen because you're suffocating, or there's something, or even locally, if some injury was creating local hypoxia, if oxygen supply to the tissues is compromised, then you're going to rely more on anaerobic glycolysis and thus glucose. Finally, astrocytes are brain cells that have a slightly different physiological purpose for lactate. Astrocytes, to specialize in their own thing, mostly use anaerobic glycolysis for energy, and that accounts for 10% of the brain's energy. And so there's always going to be some need for glucose in the brain. But they don't deliver the lactate to the liver in the Cori cycle. They deliver the lactate to neurons. And the neurons use the lactate for energy, and it helps spare their own glucose supply to use for other things like antioxidant support. So now that we've looked at the biochemical and physiological importance of anaerobic glycolysis, we've set our foundation so that in the next lesson, we can take a deep dive into the dirty practical details of the role of carbohydrates in sports performance. The audio of this lesson was generously enhanced and post-processed by Bob Devodian of Torian Mixing, giving you strong sound and dependable quality. You can find more of his work at torianonlinemixing.com. To continue watching these lessons, you can find them on my YouTube channel at youtube.com/chrismasterjohn, or on my Facebook at facebook.com/chrismasterjohn, or you can sign up for MWM Pro to get early access to content, enhanced keyword searching, self-pacing tools, downloadable audio, downloadable PDF transcripts and web transcripts, a rich array of hyperlinked further reading material, and a community with a forum for each lesson. So if you want to own these lessons and study them and really get the most out of them, then you can sign up for MWM Pro at chrismasterjohnphd.com/pro. Signing off, this is Chris Masterjohn of chrismasterjohnphd.com. You've been watching Masterclass with Masterjohn, and I will see you in the next lesson.